So the next session will really be about new drug regimens and the new DST test required. Come on in and take your seats. You ready to go? Yeah. Our first speaker is my colleague and co-chair, Daniela, who'll be talking on some of the new drug regimens and the quality control of DST. Daniela?
Thank you. And do I have... No, that's okay. Okay, so I will try to make the point in 10
Alessandra,
minutes.
could you close the doors in back?
What should I do? Okay. To make the point on the progress done in the past year on developing the laminate susceptibility testing in midget, basically. So we had signed an MTA with Otsuka, and we have received a partial reimbursement to perform this study. The study was conducted and supported somehow by the WHO, and it was conducted in the supranational reference laboratory network, and two laboratories were heavily involved in the study. That was my laboratory in Milano and Harald Hoffman laboratory in Gauting. So going to the point that the lamanide, you all know, is a nitroimidazole compound impairing the biosynthesis of the mycolic acid and therefore disrupting the metabolism of the cell wall. It is a good drug, active on both replicating and non-replicating bacilli, requires activation by the chain of enzymes that are dependent of F420 genes. And this is relevant because the mechanism of resistance seems to be related to mutation in any of the 4 or 5 genes involved. There is a spontaneous rate of resistance that was estimated in the range of 6 by 10 to the 6. The drug is well tolerated in TB patients, is associated to favorable treatment outcome and significantly lower mortality. Is not associated with clinically relevant drug-drug interaction, received a conditional approval by WHO in 2014 for MDR, selected case, and XDR TB treatment. Has received marketing authorization by the EMA and the Japanese Ministry of Health in 2014, is commercially available in UK, Germany, and many other European countries under different agreement with the local Ministry of Health. Despite the good data in support of the use of the drug, is not really in use in many countries where the drug is needed. The aim of the study was really to develop a protocol for rapid delamanide DST using the midget, that is the fastest platform for phenotypic DST that is available, and also to define a breakpoint able to discriminate between susceptibility and resistant form. So the experimental plan was to take the advantage of a set of 19 reference strains that were provided by Otsuka and evaluate how those strains, how we could correlate the MIC of this panel in REMA and midget against the reference test that was proposed by Otsuka, that is the agar-based protocol in 7H11. After that, we aim at determination of MIC distribution in both REMA and midget of clinical isolates never exposed to the drug. And the results were confirmed by the agar-based protocol. And then we explore the genetic polymorphism in the gene involved in the F420 mediated activation. So basically, the 3 methods that were compared were the standard-based agar proportion method proposed as a standard by the company, the REMA and the MIGIT. The company, during the proposal at the EMA, proposed a breakpoint of 0.2 that was kind of challenged by the UCAS committee that had asked to reduce the breakpoint. Here you can see the MIC evaluation, and you have 3 brackets. The black one is the narrow MIC evaluation that we have performed for the very large set of strain evaluated. And then you have the extended MIC that was tested for the susceptible and resistant strains. Starting from the panel of the 19 reference strains, we have challenged those against the breakpoint of 0.2 and the 7H11-based DST method, and we found 5 resistant strains with an MIC that was higher than 0.2 and 14 susceptible strains. Those results were fully concordant in the all 3 methods. And here you see the panel, and you can see that we have MIGIT1 and MIGIT2 with the data performed independently in 2 different laboratories. All the study has been conducted in parallel. When we look at the result of the VGS on the Otsuka reference panel, we found that all the 5 strains resistance were mutated in the DDN gene, and that was compatible with the results of being drug resistant in phenotypic test. We also found several mutations, both silent and not silent, leading to amino acid substitution that were apparently not linked to drug resistance. The selection of the strains was done in a very inclusive way. So we wanted to include strains from different regions as well as strains with different sensitivity pattern and with different family and different clones. We have performed the MIC in REMA and in MIGIT on 288 strains in REMA and 221 in MIGIT, and you can see that basically all the strains except for 4 were tested sensitive with a very low MIC in both MIGIT, the second table, and REMA, the first table. The 4 strains that tested resistant with an MIC of greater than 1 were strains that were independently collected in the 2 different laboratories. So 2 were coming from Gauting and 2 were coming from Milano, were isolated in Milano. Those strains were never exposed to the drugs. And here you see the extended MIC in REMA and in MIGIT for a subset of 119 strains for which the MIC was done in a very extended way. And what you can see is that there is no really low resistance to the drug. So the strains are either very sensitive with a very low MIC or very resistant. We have a slight shift of the MIC in MIGIT as sometimes is expected with the testing in MIGIT. But what we can see is that the ECOF is definitely very low, much lower than 0.2 that was proposed originally by Otsuka. When we did the correlation of MIGIT and REMA on the whole pool of strains, we found a perfect correlation between the 2 liquid testing. When we characterized the 4 delamanid-resistant strains, we found that all 4 of them were a member of the subfamily W148 of the Beijing clone, and 3 of them harbored a stop codon mutation in DDN and 1 in FBIA. When we look at all the collection that we have tested with the extensive MIC, we saw that other nucleotide changes are present in the 5 genes, but those were not correlating at this point with our phenotypic results. How are we going to progress? We are now going to progress with a multi-center study for the validation of the breakpoint, and as per the original agreement, the panel of MIGIT was including 25 reference isolates, has been sent to 6 supranational laboratories. And after that, the laboratory will be validated, and we will be asking to them to perform at the breakpoint testing in MIGIT using 75 clinical isolated per center. In conclusion, the DST for the lamanid could be performed successfully in MIGIT and REMA with results comparable to the proposed protocol by Otsuka. We are proposing 0.125, so 0.12, as a breakpoint to screen for the lamanid sensitivity. The pre-exposure high-level resistance was observed on clinical strains never exposed to the drug, so this is a concern. Low-level resistance was not observed at this point, but I need to remember you that those strains were never exposed to the drug. The full genome sequencing analysis in genes involved in the activation pathways show presence of several SNPs, not always related to an increased MIC. The stop codons in the same genes are instead clearly associated to high level of the drug. And with that, I'd like to acknowledge the great support from Otsuka and Becton Dickinson, my collaborator at the San Raffaele Scientific Institute, the group of Harald Hoffman in Gauting, the group of Stefan Niemann at the Research Center in Borstel, and the SRL network, and in particular the European SRLs that are going to be included in the second phase of the study. And then after that, we will move outside Europe. And the WHO LDR that has been supported the study from the very beginning. Thank you very much.
Thank you, Daniela. As before, we'll have time at the end after all 3 speakers have presented for questions. So I'd like to invite our next speaker, who's Leen Rigouts from Antwerp, who will be speaking on the introduction of laboratory-based DST for bedaquiline.
Good morning. Following the presentation of Daniela on delamanid, I will do present similar things for bedaquiline. So, as you know, bedaquiline, also called Sirturo, has been described already in 2005, and it's a diaryl quinolone, and it inhibits ATPase synthesis enzyme. And the molecular target, the basic molecular target for this drug is the ATPE gene that's encoding for this synthase enzyme. Also, bedaquiline has been conditionally approved by FDA for adult MDR-TB treatment, and after the phase 2 clinical trial was conducted. As you probably all know, there has been cross-resistance observed between bedaquiline and clofazimine being an old drug that has been revived for MDR-TB treatment, and this was shown in multiple publications already. What are the mechanisms behind? So the main activity of bedaquiline is acting on the energy system of the bacilli. And so the mutations seen in the ATPE gene cause high-level resistance. whereas the cross-resistance to clofazimine is related to an efflux pump mechanism encoded by the RV0678 gene, and this is leading to low-level resistance, yeah, for bedaquiline. And basically, this RV0678 is encoding for a suppressor for the efflux pump. So if you have a mutation in this suppressor gene, you will have more of the efflux pump being active and less bedaquiline being available inside the cell. That's the mechanism behind. So what I will talk mostly about now is the phenotypic drug susceptibility testing. And first data that were shown were actually already in the 2005 paper from Koen Andries. And they have done MIC testing on agar medium and in the BACTEC, the radiometric system, which is no longer available now. And they used reference strains and clinical isolates. These clinical isolates had resistance to first-line drugs or were fully susceptible, and the range observed was between 0.03 and 0.12 microgram per milliliter, which is quite low. And other data on DST is still very scarce, at least in publications. The data from the phase 2 clinical trial shows small data on the minimal inhibitory concentrations for bedaquiline, and in this paper you can also see that DMSO is used to dissolve the drug in the stock solutions, and working solutions can be done in distilled water. The reference strains used usually in these sets are the H37RV, of course, but also the BK12, which is one of the in vitro selected drug-resistant strains described in the original 2005 paper from Koen Andries. The medium used here again is 7H11 agar medium, and the conditions used, maybe I'm going a little bit too technical, but I think there's a huge demand for these data. So it's the classic standard incubation, having this CO2. What is most importantly is that you use polystyrene tubes. If you use polypropylene tubes, there is a risk that the drug will bind to the tubes, as bedaquiline is very much binding to proteins. So you can have very variable results and higher MIC. So you should use polystyrene tubes. If you use tubes or if you have plates, these are in polystyrene. An alternative method that was also used for the delaminate evaluation is the REMA, the Resazurin microtiter assay, which has been described before by Palomino and others. This is a redox reduction colorimetric method. From these data, the UCAST, which is the European Community on Antimicrobial Susceptibility Testing, they have on their webpage, you can see some recommendations for M. tuberculosis testing, and actually the only one for TB in there is on delamanate and bedaquiline. The old drugs are not even mentioned. And for bedaquiline, they have proposed 2 breakpoints. The 0.25 for bedaquiline. And this is from the agar-based data. So as MGIT is widely used for DST, there was this question, how does bedaquiline testing perform in MGIT testing? And so that's why we performed a test to evaluate the performance of this MGIT 960 system for bedaquiline against susceptibility in tuberculosis. And Gabriela Torrea coordinated this work. She's in the audience. And what was done is we first determined the QC range. It's to check how solid is this testing medium to do this test. And for that purpose, we used the H37RV strain, the reference strain, and we tested it on 10 different days using 3 different batches of MGIT and different stock solutions of bedaquiline, so all conditions were there. And you can see that the range is quite narrow. More than 95% of the tests had less than 1 dilution difference, and the mode was 0.25 microgram per milliliter. And then we determined the ECOF. As you know, the ECOF is epidemiological cutoff value for the wild type MIC value, identifying the upper limit for the wild type population. And the ECOF in MGIT, which is the red part, the ECOF in MGIT turned out to be 1 microgram per milliliter. And in this slide, all testing was done in parallel in MGIT and on 7H11. And we tested 45 probably susceptible strains that were mean strains that had never been exposed to bedaquiline before, nor to clofazimine, to our knowledge. And they had different profiles for resistance against first-line and second-line drugs. But as you can see, the ECOF on agar medium is lower than the ECOF in MGIT. It's about a 4-fold difference that you can observe between MGIT and agar. The reason for that is not very clear. As I said, bedaquiline has some protein binding. Basically, the media 7H9, which is used in MGIT, and 7H11 are not that different. Also, the supplement that you add is quite similar, so there must be something else that is binding the powder. Nevertheless, given the good QC range, we know that it is a stable method to test bedaquiline. So to further test the accuracy or the trueness of the system, we tested 74 probable susceptible strains, different from the first ones, and then 18 probable resistant strains. And which strains did we use for that? We included this famous BK12 that was described originally, but also we included some in vitro selected clofazimine resistant strains that were generated in our lab by a PhD student. And we did parallel testing for all strains included, again on 7H11 agar and on MGIT. The red ones represent the probably resistant strains, and the blue ones are the probably susceptible strains. And you can see that we have a nice bimodal curve for both systems. So the susceptible ones and the resistant ones are separated both on agar and in MGIT. And again, you can see a difference in the ECOF, the 0.25 for agar, whereas the MGIT has a 4-fold higher ECOF. However, the breakpoints are not that far away, or at least the probable susceptible strains and the probable resistant strains, their MICs are quite close to the breakpoint, which might be a risk of having false resistant, false susceptible if the test is not done very precisely. Furthermore, we looked at reproducibility and repeatability, which means that you look at inter-run and intra-run variability. What did we do? We had 5 strains, and we tested them in different weeks by different operators, and we also tested within the same day using the same set. So at the end, we found that we had 100% reproducibility and repeatability. All had less than 1 dilution difference in their MIC, which is within the range of acceptability. So the conclusion from this paper was that the MGIT seems to be a reproducible and accurate test to determine the susceptibility to bedaquiline as compared to agar medium. We determined the ECOF and the breakpoint, which turned out to be 1 for MGIT and 4-fold lower for 7H11. And as I told, the MIC values of susceptible strains and resistant strains are close to the ECOF, to the cutoff, which might be problematic if not done carefully. But further studies are needed on clinical data are needed from a larger number of susceptible and MDR-TB strains to finally set the clinical breakpoint. What does it mean for the outcome of the patients? That was not addressed in this study. There have been more data on MGIT. The study done by Keller and colleagues, they used MGIT and the TB-Exist software. The main difference probably with the largest study done in our laboratory is that they didn't have the powder available when doing this test, so they used bedaquiline tablets and they had to mince them, and then they calculated what was the amount of active compound. So there might be some bias even if they tried hard to do their best. What they have shown is that in their system, they did not exactly test the same dilutions, but they found an ECOF of 1.6, which is a little bit higher than what we found, but that could be due to the dilutions that were used. And also in their strains that they tested pan-susceptible strains and MDR and XDR strains. They did not find a significant difference between the medians of these groups of strains showing various first-line drug resistance or second-line drug resistance, which was also not seen in our study, by the way. And also they had some stability tests done and couldn't find problems on that issue. There have been two multicenter studies done by Daniela's group and others, and where 8 different laboratories were involved. They compared 7H10, 7H11 agar, and 7H9 broth, and using only the H37RV strain. So it's kind of QC data that they were gathered, and they found similar ranges of the QC data for H37RV. Daniela, I think the 7H9 broth was not the MJIT, but another system. Is that correct? So that might explain the difference with the MJIT compared to the 7H9 broth as in REMA. So we have some additional, that's about all there is on data in literature at this moment. So another important lesson to take home is that Löwenstein is not suitable for determining bedaquiline resistance, as there is a high protein binding, unless if you go for very high concentrations, like is the case for rifampicin with 40 microgram per milliliter versus 1 microgram in MJIT, but better to avoid this. Stability of stock solutions, we have done that in our lab, and we found that up to 3 months they can be stored at -20 degrees. And in REMA plates, or once dissolved in lower dilutions in broth in general, maximum 2 weeks of storage in the fridge. So a little bit of summary of the phenotypic DST. What is important that you have the difference in ECOF and breakpoint between agar testing and MJIT, and QC ranges for the reference strains, of course, are in line with that. So now there is, of course, a rollout of use of bedaquiline in many sites and countries, and labs are wondering how should we test bedaquiline in our labs, what can we do? J&J has arranged with NIH to procure the powder through them. So basically you have to go to the NIH page on the AIDS reagent ordering, and there you can order the bedaquiline powder, so the pure powder to do testing in your lab. How can you have good reference strains? Of course, you can use the H37RV as a control strain, what you use for all your strains. And in our lab, we are managing a public collection of mycobacterial strains, and J&J has deposited the bedaquiline-resistant reference strain in our lab, and we have also deposited the clofazimine-resistant reference strains. I left in the back some information for those who are interested to obtain these strains. Even if you implement, there remain some challenges in...
Leen, just one more minute, please.
This is the last slide, yeah. So the remaining challenges is that bedaquiline testing is not yet included in EQA panel, proficiency panel testing. And regarding the molecular tests, I did not address it here. There is other presentations that will be done by Koen Andries later on in this conference. And there's also the poster session by Gabriela Torrea, where you have more details on MJIT bedaquiline. But it is clear that there is a lot of mutations appearing in the RV0678. That's the efflux pump-based mechanism causing cross-resistance. But it's still unclear what is the related association with the level of resistance. There is as well clearly mutations that are not associated with resistance either. And as we come across a lot of variable mutations in this gene, it will be very difficult to develop a rapid molecular diagnostic test for bedaquiline testing. Maybe the ATPE mutants will be more straightforward with a higher level of MIC and higher clinical significance. We don't know yet. And we have not come across these mutants in clinical isolates so far. So, thank you.
Just to clarify, the reproducibility study was the one that was set up by the Janssen to check the reproducibility, and I presented the preliminary result at the WHO in preparation for the meeting in March.
Great. Thank you, Leen. Our final speaker for this part of the symposium is Claudio Köser from the University of Cambridge. He'll be speaking about genetic diversity in the development of new DST.
Thank you very much to the organizers for inviting me to give this talk, which I think is going to link quite nicely what we've heard just now about DST and the talk that we had in the first session about elucidating the genotype to phenotype relationship. I've changed the title slightly just to make it a little more informative. So I'm going to talk about the implication of the genetic diversity both within the Mycobacterium tuberculosis complex and M-Canetti on the development of new DST assays. And this is an overview of my talk. I'm going to quickly talk about the importance of resistance mechanisms. I'm going to pick up on some of the things that Derek said, but maybe explain them in a slightly different way. Then we're going to talk about the consequence of genetic diversity, both in terms of resulting in systematic false positive results with the for fluoroquinolones, for the Hain genotype second line test, both as a result of non-synonymous mutations, but also synonymous mutation. And then I'm going to talk about perclozone. So both of these studies sort of work in progress, so I welcome your comments. Particularly, I'd like to talk to any of you who treats TB in Russia or has links to policy making in Russia regarding to talk to you about perclozone. So I've got to put up this disclaimer, so the views expressed in this talk are my own and not necessarily those of the UK Department of Health or Wellcome Trust. I've received some funds from Janssen and Gates in the past, and I've received an award which is sponsored by Hain, and I'm a find consultant. A lot of different people have contributed to this study. Some of you are in this room and don't have time to mention all of them, but I would like to thank Matthias Merker in particular, and also Erika Lessem from the Treatment Action Group in New York. If you're interested in the whole issue of resistance mechanisms, we published an opinion piece in The Lancet earlier this year, which consists of two parts. In the first part, we essentially argued that the elucidation of resistance mechanisms should be mandated in order for new drugs to be approved, just in the way that they are mandatory for new antiretrovirals. And if you want to read this article, I think it's actually at the back. And in the second half, we analyzed the consequences of some of the recent rulings on gene patenting on TB resistance mechanisms. Now, I think elucidating resistance mechanisms early on during drug development is really important for a number of reasons. And I think resistance mechanisms can essentially be broken down into three different classes. The first is resistance genes that are always essential. So examples would be RPOB, NHA, RPSL, or RS, because here evolution is on our side. So essentially only very few resistance mutations are possible. And as a result, the likelihood of elucidating the genetic basis of resistance comprehensively is very high, just because we have very few variants that are possible. We then have sort of this intermediate category where these genes are non-essential, but in patients are sort of pseudo-essential. And the best example here is CAG. So if you select for it in vitro, you get lots of different mutations. In the clinic, however, you have some dominant mutations because these have a higher fitness and therefore spread more readily. And so again, finding out what these dominant mutations are is easy, but then you have lots of rare mutations where we, I think, are going to have trouble to find what they all are. And then we have the last class. We have these non-essential genes. The best example of PNCA. And just to give you, I did a quick calculation. So PNCA has 185 amino acids, so excluding the start codon and stop codon. And so as a result, if we just look at single codon changes, we have, so that's just 128 times 23, we have over 4,000 single nucleotide, single codon changes that are possible. This excludes stop codons. So not all of these mutations will cause resistance, but so it's really hard to study all of them, because as you sequence more and more strains, you'll find more and more new mutations. And so therefore, these are sort of what Derek referred to as the Achilles heels in terms of resistant genes and finding what the mechanism is. Linked to that is really the question of what kind of assay, genotypic assay, can we develop to detect these mechanisms. So for essential genes, even assays that only target small loci will have a high sensitivity for these essential genes. For these sort of intermediate genes, they are probably still going to have high sensitivity if we target these dominant mutations. But for these non-essential genes, unless we sort of take a shortcut and say, okay, let's assume that any mutation that we detect in PNCA causes resistance, we're going to have some trouble with the kind of assays that we have. And this is the slide that Derek showed you early on. So this is basically different resistance, different antibiotics at the bottom, and then different resistance mutations. And these bars shows how often these mutations occur. But I changed this and adapted the slide slightly. And at the bottom here, you have color-coded the different genes or mutations according to the class of resistance. So you can see here that we're quite lucky that for isoniazid, rifampicin, and ethambutyl, we only have resistance genes that are either always essential or sort of pseudo-essential. And the best example here is this CUT-G315 mutation that is very fit and as a result very frequent. Now, but then when we go further to the right, it becomes more difficult. So pyrazinamide, we have one essential gene here is RPSA, but this is actually a mistake, this is false association. But so you can have PNCA, you have lots of very rare variants. The only reason why this mutation here is frequent, that's a change at codon 57, and that's the mutation that's responsible for intrinsic resistance of Bovis. So this is deeply rooted in the phylogeny. As a result, it's frequent. The other reason why this mutation here is frequent is a promoter mutation. This is present in one of the clones in Eastern Europe, which just spreads everywhere, and this again is the reason why it's more frequent. But so you have these large number of very rare variants, and then for streptomycin you have a combination of essential gene and non-essential genes. The other reason why this resistance mechanism is essential is because this really dictates how likely it is that we have intrinsic resistance in a significant proportion of the population. Now, for essential genes, this is very unlikely because these mutations will usually incur a fitness cost on the strain unless you have compensatory mutations. So in the absence of selection, even if these strains do occur, they shouldn't spread particularly well. For these intermediate genes, I think you have an intermediate chance. For non-essential gene, in my view, it's not a question of whether there are intrinsically resistant strains or not. The questions is how frequent they are, and this depends on two things. The first is how deeply rooted this mutation is within the phylogeny, and also how fit these particular strains are. So the best illustration of this is, I can show here. So this is a tree that shows our current understanding of the population structure of pathogens that cause TB. So you have M. candida at the top, M. tuberculosis complex down here. And so we have intrinsic pyrazinamide resistance down here that is very old, but fortunately M. bovis and BCG are obviously not widespread globally, certainly not compared to mycobacterium tuberculosis. Then we have intrinsic resistance to cycloserine and M. bovis. We have intrinsic resistance to isoniazid and ethionamide in more recent BCG strains. And then we have, again, ancient intrinsic resistance to pyrazinamide in some, if not all, M. candida strains, and this also extends to P824. And we're currently investigating whether this also extends to delaminate and TBA354. Now, the next thing I want to talk about is the high-end second-line assay. You're probably all aware of how this works. So this targets a small area in GRA, and the assay has two different kinds of bands. It has wild-type bands, and it has mutant bands, which are not shown here, but they show but what I did show is the kind of mutations which can be picked up. And you'd interpret this particular assay as being resistant either if you get binding of mutant bands or if the wild-type bands do not bind. Now, the only systematic error that had been described to date was due to this double mutation. It's a TATA and 90G mutation, and it's been shown conclusively by different laboratories that this double mutation doesn't confer resistance in effect and seems to confer hypersusceptibility to fluoroquinolones. But the problem is that this mutation here at codon 90 prevents the wild-type 2 band from binding. As a result, you have a systematic false positives. But it had been unclear whether this mutation was monopara or polyphyletic and how widespread these particular double mutations were. But we did have some indirect evidence. First of all, these double mutations seem to be exquisitely rare. So in my own internal database, it was only described in 8 out of 200, over 200 studies that looked at GRA mutations. We also know that this TATA mutation seemed to be a marker of the Uganda genotype. So this suggested that maybe this double mutation is found is a subgroup of the Uganda genotype. And the other piece of indirect evidence was that there are 2 recent studies that are done in the Congo that showed the highest frequency of this particular mutation. So in these 2 particular studies, 7% and 60% of MDR strains had this double mutation. And we've screened over 5,000 genomes to look for this double mutation, and then indeed found that, and then zoomed in again on the Uganda genotype. And this is a maximum likelihood tree with so 87 Uganda genotype strains with 2 outgroups, strains that are Uganda strains but don't have the mutation. Now all of these strains have the TATA mutation, but then you can see here that a subgroup within Uganda, as I sort of predicted in a review that I did a couple of years ago, have this double mutation. So it really does seem to be sort of a subgroup within Uganda. Now the other interesting thing that's worth noting is that within this Uganda set, you had a strain that had the TATA mutation, but also had the A90C mutation. This change had never been described in the literature before, and the reason for this is probably because this is a dinucleotide change in the same codon relative to H37RV, which is very unlikely to arise, but it's only a single nucleotide change relative to the A90G mutation. And we're currently investigating whether this caused resistance, and this could be sort of a candidate of a sort of genotype-specific resistance mutation. So the upshot of this is that the Hain assay is probably going to have very poor positive predictive value in Uganda and the Congo. Now the other thing we looked at was synonymous mutation. I think it's now well described, certainly for RPOB, that synonymous mutations can cause to systematic false positives, but so far these hadn't been described in GRA. And again, we looked about 5,000 strains, and we only found 4 synonymous mutations that cause systematic false positives. So here are the mutations, and I just listed the bands that don't bind if these mutations are present. All of these were very rare, at least in our data set, which wasn't really representative, with the exception of this last mutation here that was found in 8% of MDR strains in Medellin in Colombia. So again, in this particular setting, the Hain assay would have a poor positive predictive value. The interesting thing is that we also found synonymous mutation didn't cause systematic false positives. And the most interesting mutation is maybe this one here. So you can see that different synonymous mutation in the same codon can either lead to false positives, sometimes not. And I think the reason for that is that this particular mutation here is right at the very edge of the band, whereas this mutation here is sort of further in the middle and therefore has probably got a bigger effect. And at the moment, I'm sort of conducting sort of some follow-up experiment where I had some plasmids synthesized with different combinations of these genes, because I wanted to investigate what happens if you have, for instance, this mutation here in combination with a bona fide resistance mutation, say this one here. And this particularly, if you have these mutations together, not only does the wild-type band not bind, but also the mutant band doesn't bind. So really this illustrates that unless you do sequencing, you can get sort of different types of errors with your probe-based assays that we hadn't really appreciated so far. Now, the last thing I want to talk about is perclozone. So this is a drug that I'd never heard about until the beginning of the year, but it's actually been approved for the treatment of MDR-TB in Russia since 2012, and it's been added to the list of vital and essential medicines in 2014. This essentially means that the access to the drug is guaranteed and the price is controlled in Russia. Now, I think I'm not going to get sued for libel if I say that the evidence for the safety and effectiveness of this drug is very limited, and that's putting it diplomatically. And there are efforts to register this drug in African countries and Commonwealth countries. Phase 4 trials are ongoing. The official guidelines in Russia by the Department of Health don't mention perclozone, but it is mentioned in the guidelines by the Russian Association of Pneumologists. And again, they advise empiric treatment. There doesn't seem to be any lab that conducted DST, with the exception of maybe one lab in Russia, and I'm doing some digging to try to get more information there. Now, the reason why I got interested in this is because of the study here on the bottom by Gopal et al. This is a group in Singapore that selected for in vitro mutants, and they had found that perclozole is a prodrug that is activated by ETHA, which is a monooxygenase, and it targets mycolic acid synthesis by targeting HAT-A. Now, the problem is that ETHA also activates ethionamide and prothionamide. It also activates thiacetazone, and actually thiacetazone has the same target as perclozole. So this has very practical implications, because according to the Russian guidelines at the moment, perclozole and ethionamide and prothionamide could be given in the same regimen, which is not a good idea if they share a resistance mechanism. Similarly, the pharmaceutical company recommends on their website this particular regimen, which contains both perclozole and prothionamide. And this is unwise, again, to put it diplomatically. But the other question which I wanted to ask is, well, prothionamide and ethionamide are widely used in Russia, so how much pre-existing resistance do we have to this drug? Thiacetazone is not going to really be an issue because it's only ever been used in Russia for leprosy. So what we did to answer this question was we looked at just over 700 MDR strains, about 400 MDR strains from Samara, just under 300 strains from Uzbekistan, and this is just Moscow up here in the top left-hand corner, just to orient you. And this is the phylogenetic tree, maximum likelihood tree. So you can see here that, as expected, the vast majority of MDR strains in this setting are Beijing strains. And the MDR epidemic in this setting is essentially due to 3 big clades, A, B, and C. And these colors here show whether an ETHA mutation is present or not. So what's really worrying is that you have, for instance, this clade here. Essentially, all of these strains have a promoter mutation. In clade C, we have this dominant mutation. And then in clade B and C, we actually have a homoplastic deletion, which is a good signal for positive selection. And actually, we've looked at some other data sets, and this promoter mutation here is homoplastic as well. You have some clade B mutations, which strains that also have this mutation, which suggests that actually these might be a resistance mutation. If we look at, when we then had a look at the DST data, because for most of these Beijing strains, we had DST data, so you had DST data for about 570 out of the 591 for these strains. And of those, 15% were wild type, 74% had a mutation, 20% had an indel or nonsense mutation, so these should be strain mutations that really cause resistance. 8 strains had this dominant homoplastic indel, 30% had these promoter mutation, and then 16% had this dominant mutation here. Now the question is, does this lead to ethionamide resistance? And the answer to that is, well, sort of. Now I've did a little statistical analysis. So basically I've plotted the rate of resistance for the different classes that you see here. And the error bars here correspond to 95% confidence intervals. So you can see here, so if you have any ETHA mutation, your chance of being either ethionamide or prothionamide resistance, and these are either results based on LJ or some midget data, increases from 20 to just under 40%, and this is statistically significant. Now, if we just look at the subgroup of indels, this increases from 20 to about 60%. But I mean, this really suggests that phenotypic testing is really poor, because I mean, we know that any nonsense mutation should lead to resistance. So essentially what this means is that phenotypic testing misses 40% of resistant strains. If we just look at the subgroup of this dominant mutation here, again, a statistically significant from wild-type strains. It has obviously got larger error bars than if we pool non-synonymous mutations. But if we look at this promoter mutation and this other dominant mutation, we don't find statistically significant difference in error rates to the wild type. Now, personally, I don't trust any of this phenotypic data, but I mean, it seems to be broadly right. So what we're doing at the moment is we've selected strains from these dominant resistant clones, and I'm almost done, don't worry. So we've taken strains from the promoter mutation, from this 314 mutations, and the homoplastic mutation. Both strains that tested resistant initially and susceptible, and we are, based on the tree, picking wild-type strains that are as closely related as possible to have sort of perfect, as close we can get to an in vitro selection experiment. And where we have mutations that are homoplastic, we'll pick mutations from both groups. So, for instance, from this mutation here, we're going to pick strains from here and here. And we're about to start to do MSCs for ethionamide. Now, we'd ideally, obviously, also like to do this for perclozone, but the trouble is getting access to the drug. So if any of you can get hold of some drug, please come and see me. But this really illustrates that even if this drug is safe and effective, which is an open question, it might have very little use in this particular setting just because of the pre-existing rates of resistance, which is why I think it's so good that the WHO is switching to whole genome sequencing for surveillance, because you can use these data to do all sorts of things. Thank you very much for your attention.
So we're running about 8 or 9 minutes behind schedule, so we probably have time for one burning question for the 3 speakers from this session. Otherwise, we'll ask you to grab them after the end of the symposium and speak to them directly before we move on to the next panel. Are there any specific questions for Claudio or Leen or Daniela about their presentations? Or I'd like to thank the panel and the 3 speakers. Leen has a question. Okay.
For yourself.
Yeah.
I just have a clarification regarding the bedaquiline MGIT results for the MICs for the resistant strains. These were all RV0678 mutations mostly, so the ATPE mutants might be much higher. So just to keep that in mind.
One question.
One quick question.
Yes, to Daniela, about these 4 delaminate resistant strains, which appear to be naturally resistant. So how much is known about these? I mean, notice that 3 have the same mutation. That might be a part of a cluster or something.
There is this possibility. So all of them are part, by sequencing, are part of the same subclone, Beijing subclone. 3 of them are harboring the same mutation. We are now investigating the possibility. They are isolated in 2 different laboratories, so the strains came independently in 2 different laboratories in Europe for different reasons. And we are now going back to the original data and investigating. 2 of them may be more closely related. The 3rd one is related, but it seems to be an independent strain. Could have been a cluster originally. The 4th one is a different mutation, still the same subclone, isolated from a patient from a completely different Eastern European countries. It still is close, but is definitely not part of the clone.